Okay, so thank you very much for the invitation. As you just heard, my topic will be strategies which might be best for jawbone tissue engineering using stem cells. The audience might be aware that there's quite a lot of different types of stem cells within the jawbone and within the teeth, as you can see here on that slide. And without going into detail, the difficulty with these particular stem cells is that you might not have them in the time period when you want to use them within the patient, and that their availability is quite limited. Therefore, we wanted to compare them to mesenchymal stem cells, which have quite a good potential. So they can differentiate towards osteoblasts, and they can differentiate towards endothelial cells and smooth muscle cells. So they have the potential to do everything which might be needed within
post-tissue-derived stem cells. And just to show, as compared to what we did before, that we can also do that with the natural ligands such as ATP, UTP, or UDP. And if you compare the first two squares, which is the control and the differentiation towards endothelial cells shown by Enos, I think you can see using the ligands that there to some extent is an enhancement or actually a loss of the stainings of Enos. For example, the loss can be shown for UDP on the right bottom. So if we summarize this, we can come up with that scheme, which you can see on the left side. So there are several cholinergic receptors where the upregulation pushes mesenchymal stem cells towards the endothelial lineage, and actually one Y6 where the downregulation enhances endothelial cell differentiation. Well, of course, thinking about angiogenesis, especially the functions such as branching plays a major role. And here you can see an example where we differentiated adipose tissue-derived stem cells towards endothelial lineage. And on the very right side, we used a specific Y1 agonist. And I think you can see that the branching got more as compared to the controls in the middle and on the left. So therefore, we summarize that P2Y1 receptor agonists can improve endothelial cell sprouting. We did a similar experiment for the smooth muscle cell differentiation. Here our readout is calponin, which we showed you before, is a specific marker for smooth muscle cells, also in natural primary cells. We looked for specific markers such as calponin on the RNA and on the protein level, and for a specific staining of the cells. Now, similar to what we did before, we used artificial antagonist here for Y1 to see if we can enhance that smooth muscle cell differentiation. C, once again, is a negative control. S is a positive control with a staining for calponin. And here we showed that different amount of that specific antagonist is enhancing the staining for calponin and therefore the differentiation towards smooth muscle cells. So this is clearly functional. We did the same actually with the agonist. So therefore, of course, we expected to lose the staining for calponin, and I think you can see that here on the left bottom. So, of course, we've been interested how cholinergic receptor signaling is enhancing endothelial cell differentiation. So we've been using an array and looked for different signal transduction pathway. And to make a long story short, I can show you here's a scheme, one on the right side for the endothelial cells and on the left side for the smooth muscle cells. And we say that for the smooth muscle cell differentiation, specifically the signaling via ACT and NF-kappa B plays a major role, whereas for the endothelial cell differentiation, the MAP kinase P38 upregulation plays a major role if cholinergic receptors are influencing endothelial cell differentiation. So I already showed you the upper part of our suggestion how cholinergic receptors are playing a role in the differentiation process. And I can add now the two other lineages. So we have one pair of cholinergic receptors where the downregulation of Y4 and Y14 pushes the cells towards the osteo and adipogenic finish, and the upregulation of the same pair of purinergic receptor pushes the cells towards endothelial and smooth muscle cells. Then we have one single receptor where the downregulation X6 pushes the cells towards osteoblast, and the upregulation pushes the cells towards adipocytes. And we have the same for endothelial and smooth muscle cells, where Y1 upregulation pushes the cells towards endothelial cells and the downregulation to smooth muscle cells. And further on, we have specific markers which can enhance that differentiation process. Let me summarize that. We suggest a combined model of osteogenesis and adipogenesis. So first, put stem cells on a biomaterial and put that in an incubator together with bone-specific purinergic ligands. Why? Because the bone cells will be in a very tight environment later on and cannot be released. So therefore, that has to be done in advance. Then you can use that biomaterial, which is loaded now with stem cells differentiated towards osteoblasts, and put that in a bone defect. And if you then need angiogenesis as well, you can use endothelial-specific purinergic ligands from the outside to have endothelial sprouting from the bone material into that artificial tissue. If I summarize throughout what I just told you, there's a lot of things you have to consider if you use such a material for research or actually in therapy. So you can use dental material if you just want to go towards hard tissue such as jaw bone. You have to digest that and expand that, and from the dental cells, you can differentiate them towards osteoblasts. But if you need also endothelial cells, you need mesenchymal stem cells to do this due to their pre-commitment towards hard tissues. You can add soluble factors such as ligands for purinergic receptor to enhance that function. Well, then you can go towards preclinical studies in animal models or freeze those cells for further use. If you think about possible clinical applications, you can use a biomaterial which is loaded of these cells, as I just shown you before, into that material. You can also use dental implants, so you a scaffold used with these factors to regenerate bone, and maybe in the future even go for tooth regeneration. However, I have to say that in the EU, hurdles to use material which left surgery for expansion and differentiation is huge. And this is the end of my talk. I want to thank my lab, especially Nina Zippel and Yu Zhang, which have been PhD students in my lab. And on the right side, you can see my major cooperation partners. And with this, I thank you for your attention.